So our next speaker this afternoon is Luis Brito. He will be discussing as part 2 of this session, genotype by environment interaction. As you know, Dr. Brito is at Purdue University, clearly one of the important contributors to our project. Over to you, Luis.
Good afternoon, everyone. Very nice to meet many of you guys here and visit with others who I have met before. So we are going to describe a little bit about genotype by environment interaction. There has been already some discussions here, and you guys know better than me that sheep in the US is raised across a wide range of environments, climates, and management system. So, for example, some producers may give more assistance during lambings, others don't. So there is a wide variety of management and environment where our sheep is being raised. For example, if we look here, the average of some climatic variables, so this map shows different it shows different ecoregions. So it's a map of the US. We can see the average temperature, rainfall, and elevation across all those regions. They differ. And when we look here in this map, that shows the location of some of the flocks that are contributing to our project, we see that they are well distributed across all those different ecoregions or different environmental conditions. Another challenge that we have to keep in mind is climate change. So there has been recent reports that shows that the past 10 years have been the hottest on record. There are some regions, including North America, where we can see that some areas they are warming faster than the global average. So considering all these different management conditions, different environments, and also climate change, there is a need for us to breed more robust and resilient animals. I think it is important that we define what it means. So robustness is the animal's ability to remain healthy and productive within a defined environment or set of management conditions. And climatic resilience can be defined as the animal's ability to either be little affected by or capable of quickly recovering from changes in climatic conditions. And as we all know, so the phenotype, or what we measure on an individual, it depends on their genetics, it depends on the environment in which those individuals are being raised. And one point that we often forget is genotype by environment interaction. So in other words, the best genotype for one environment may not be the best for another environment. And you may be wondering, what do I mean by genotype? So genotype can be different breeds, it may be different breed combinations, it may be biological types, for example, large versus smaller animals, it may be the animal themselves or sire families, and it may be animals from a certain breed that were being raised for generations in different states or geographical regions. Now, what is the environment? So what do we mean by environment? Environment is everything that is not genetic. So it could be climate, it could be the level of nutrition, so the quality, the availability, there is management, difference in pastures, also different disease, for example, parasite challenge. So there will be certain regions where there is much more challenge, let's say, to gastrointestinal parasite than others. And also, as Hilal already presented here, the management practices survey, that is a very good way for us to characterize different environmental parameters that we can use for understanding this environment. And so when we look at genotype by environment interaction, this is a nice way of representing it. So on one side, we have the environmental value. So that is basically, you can imagine, for example, the level of assistance that you provide during lambing, or the average temperature, or humidity, rainfall, any other environmental variable that we have talked about. On the Y, on the vertical, we have the phenotypic value. That could be survival, it could be body weight, or any other trait that we are measuring. And each color represents, let's say, a sire family. When we see, for example, on the two plots on top, we can see that there is equal or no change across the environment between the different environments. So, for example, in the two plots on top, the green sire family is always the best, then the blue, then the orange. The ones on the bottom are the ones in which there is a genotype by environment interaction. And those are two types of genotype by environment interaction. One of those, it is the less severe, in which there is no real ranking. In other words, when at a certain level of the environment, the difference between the three genotypes is smaller. At the other range of the environmental conditions, the difference between the genotypes are much larger. However, so even though there is an interaction, it is not as severe as the one that you have on your right in the bottom, where you can see on the lower environment, for example, let's say in colder conditions or in lower rainfall, the blue genotype is the best. Then on the opposite environmental condition, the green genotype is the best. So that is a clear example of a genotype by environment interaction, okay? Here is just other examples where we have two environments, and then we can see in this case there is no genotype by environment interaction, okay? And when we say that there is no environmental genotype by environment interaction, it doesn't mean that the performance is the same. As Dr. Lewis mentioned earlier here about other health, so it could be that, for example, if there is indeed no genotype by environment interaction, it could be that in one environment or under certain conditions, all of them have, let's say, higher other health, and then they both equally decrease in another environment. So as I show in the bottom here, it doesn't mean that the genotypes will have the same performance across environments. It just means that the difference between them will be consistent if there is no genotype by environment interaction. And here, as we already saw, this means that we have significant genotype by environment interaction. Here is just one example where we have three environments. In the one on your left, that means most of the year there is a warm environment. In the one in the middle, you have both warm and cold. The one on your right, it is mostly cold. We have here two sire families, or you can also think of two breeds, in which the B is well adapted to cope with heat stress. The A is well adapted to cope with cold. Then we can see in the mostly warm conditions, we can see that the performance of B in the more yellow, that means more warmer conditions, it is the highest. Then as it gets colder, the difference between A and B will be smaller in this left plot, because in this environment, it is mainly warm conditions. So the B will perform better across most of the time. While on the other side, when it is mostly cold, then what we see is when the temperatures are, let's say, lower, or warmer, the B will perform better than A. And as the temperatures become colder and colder, the genotype A that copes better with colder temperatures will perform better, okay? So what this shows us is, depending on the environmental condition, one genotype will perform better than the other. And what are the implications of these genotype by environment interactions for a sheep breeder? So first, Some breeders, they are better adapted to the resources that the environment provides. So if we know this, we can then decide which breed will be a better fit to a certain climatic condition, nutrition level, and management practice. Secondly, as we change either the genotype or the environment, we may need to alter the order. So for example, if you change which breed you are raising, or if you are making genetic progress in your populations, you may need to change the level of nutrition and management that you provide to your individuals. Or if you do change, let's say, nutrition and management, you may need to get animals of higher genetic merit or a breed that is more productive, that is more suitable to that higher nutrition and environment that you are providing. A third implication would be, let's say, if we have certain breeds that are more adapted to a certain environmental condition, when you are buying replacement animals, not necessarily they will adapt to that new environment that you are going to be raising them. So it is important for us to be aware of that. Lastly, we need also, when we have a certain genetics, we need to use this information to determine what will be the best environment for them. So for example, if you know that you have a certain breed that is not very resistant to parasites, that may not be the best breed for you to raise there, or you may need to adopt more parasite control measures so that the individuals don't suffer much, okay? And when we are measuring genotype by environment interaction, there are a couple characteristics that we need to take into consideration. So as we are collecting the data or defining which data is more useful for us, these are some points that I want to emphasize. First, it is very important that we have sires with offspring across multiple environments. So we need to have sires that will have offspring distributed across the different levels of the environment. We need large phenotypic data sets. As Dr. Lewis already mentioned here, in the genomics era, phenotype's king, right? So we need to continue collecting phenotypic data. It is also important for us to standardize data collection protocols, because a lot of times we may find difference across environments or states, and it may not necessarily be a genotype by environment interaction, but it could just be that producers are measuring traits in a different way. Also, we need to have good characterization of the environment. So it is by doing surveys, is it by recording different phenotypic conditions? So we need to characterize the environment in a good way. And for us to quantify GBI, there are basically two main methods. One is that we call multi-trait model, where we measure a trait in each environment. We consider the measurements in each environment as a different trait, and then we see what is the genetic correlation between them. So for example, litter size measured in, let's say, out west or versus east, for instance, and then we see what is the genetic correlation between those two. If it is a high genetic correlation, that means there is no GBI. If it is lower, there is GBI. Another approach is called reaction norms, and it is basically one way that we can plot the genetic value for each individual across the environmental gradients. So instead of having a single EBV, the individual could have one EBV in each environmental gradient. And that can help us to assess the level of GBI. Now, just to finish up, I just want to provide a couple slides here that were actually prepared by Dr. Lewis on incorporating GBI in a breeding program. So imagine that we have here two genetic lines. We have line 1 on top and line 2 in the bottom. And those animals, or those two lines, they were fed basically a high energy and protein food, a medium or a low protein and energy feed. And we can see that is on the highest feeding level, which is ad libitum, which is on your left. The line 1 grew 1.19 times as fast and was 1.17 times as efficient compared to line 2. However, when the quality of the feed was low, we see there was almost no difference between the two lines. So this tells us that there is basically a substantial genotype by environment interaction in this case here. And the sire 1 progeny, they are much more sensitive to the environmental challenge as compared to the sire line 2. So you can see that the first one decreases much faster as compared to the other one. So we can basically obtain a slope, and then we can select which individuals are less sensitive to these environmental changes. And one quick update here. So this is a work that one student from Dr. Lewis did, where he basically wanted to combine climatic and management information to define production environments, or as we call, eco-management clusters. After he defined these eco-management clusters, he also had data from performance for 3 main traits, from 9-day weight, fecal egg count, and Fibonacci score. And then based on the data from the management information that he obtained, plus these phenotypic measurements, he was able to use this cluster information to assess if there was genotype by environment interaction for these traits. And he assessed sire by environment interactions, and what he observed is that there is significant difference on the variance of those traits. Here we are showing fecal egg count across the eco-management clusters. In other words, in some of those there is much more variability as compared with the others, and the sire by eco-management cluster interactions, they explained 12% for fecal egg count and close to 20% for body weight of the phenotypic variation, indicating that there is substantial genotype by environment interaction for these traits, body weight and fecal egg count, and this should be considered in a breeding program. So as final considerations, this is an area that we are continuing to work, so we are assessing genotype by environment interaction for different traits. And as I mentioned, we do need well-structured data sets, so we need to have sires that have offspring across a wide range of environments. And G/E has been reported not only for our group, but also in other groups. And when there is G/E, the breeders should select the animals that are better adapted to the environments where their offspring will be raised, or adjust the management practices to allow the animals to express their full potential. And there are some papers that describe all of these in more details as well, and we are happy to share them afterwards with you guys. So thank you, and here are just some questions that I would like to ask you, or in case if you also have any question, feel free to ask as well. Thank you.
Question, plenty of time.
Louise, do you think, especially with some flocks that have lower numbers in the flocks, do you think that we'll have enough numbers to be able to make these meaningful G/E by M interactions?
Yes, John, and usually what we see is we have, let's say, low numbers, we do our GBI analysis, there is metrics, for example, the standard error or how accurate those estimates are. And then, so I would say we can do the analysis, and then we can look at the results and see how accurate they are. But I would say if you have, for example, a certain breed that is only being raised in one environment or under one type of environmental condition, and then you have very few individuals being raised in a different environment, then you may not identify GBI. It doesn't mean that it does not exist, but it could be you just don't have enough data to capture that interaction. So I would say the more robust the data is, the better. And then until we get there, we can also look and see how our estimates, how much we can trust on those estimates that we get. Maybe a question for you guys. Have any of you guys realized, for example, if there is a certain breed, if you may have, let's say, moved a flock, or if you sold animals to another, to be raised in another environment, or you bought animals from another environment, and you noticed that they did not adapt very well in that condition? Have you guys, any of you guys noticed any, like, substantial GBI?
All right, a lot of you probably know us, and some of you don't. We moved from southwest North Dakota to northeast Kansas about 10 years ago. Pretty big difference in environment, an area of about 15 to 17 inches of rain to one close to 40. Also went from fairly severe winters to, until this year, no winter. We found a lot more worm challenge. In North Dakota, we would worm every couple years once if we thought about it, usually with injectable ivermectin, and not because we needed to, but just because somebody in Extension told us it was important to worm. When we moved to Kansas, we end up following pretty much the same deworming protocol. The second year, we culled about 20% of our ewes, because I guess you guys would call it high DAG score. Because we didn't want to change, we didn't want to start worming every 2 weeks or every month or whatever some people were. And we found that now when we went on NSIP and started doing fecal egg counts, we do have more resistance perhaps than the breed average. One of the things we found, and it doesn't show up in any of, and the sheep do well. Rambouillet are a very adaptive breed. I mean, they're merinos, they're raised all over the world, in every environment. But going back to what you said about different animals and different flocks adapting to the environment. I mean, our sheep, I think by general observation, have gotten smaller framed. They're not as tall. The grass has a higher water level, so I think we're bigger middled, probably have a bigger rumen, and we haven't measured it. It's just observation. The biggest factor we found is our wool quality is not as good, and it's mostly a factor of environment. I mean, wool is not made to grow in a high humidity, high rainfall area, and not made to look nice. In southwest North Dakota, it's easy to make wool look good. Even bad wool looks good there because it's so dry. So that's just some of the things we found. The biggest thing was, for our move, was worm resistance, or maybe not resistance, worm availability. Because in southwest North Dakota, where there's winter for 6 months of the year, I mean, the life cycle of worms really gets interrupted. So that's some of our observations.
Thank you. That's a very nice of GBI. Yeah.
My question is this. So here we have, in the fourth case, the environmental value, we have a crossing over, an interaction. Several ways you can deal with this, right? One could be, right, if I'm in the blue line and I'm in an environment that's good, I'm happy. That fits me. That animal does better. Green line, opposite. So one would be to make selection decisions. The other would be to modify the environment to suit your animal's needs. Does that make sense? So we could change the genotype, or we could change the environment, or both for that matter. What would you do? You've shifted to an environment that is partial, right? A change. So what do? Do you change your selection strategy, or do you try to ameliorate the consequences of the environment? This becomes really important when we think about how we would go forward, because we can shift things around.
Yes.
Is that a fair question?
Yes. Is that clear?
You have to change your
Yeah.
selection. It's a lot easier to change that than environment.
Is that the general view? People would revise their breeding decisions, the rams they buy, to fit their environment? Or would they try to change the environment to better fit the kind of animal they have?
Depends on your resources. I mean, if you needed more nutrition to handle the parasites and corn was cheap, you could add corn, and every bite of corn is a bite without parasites. I think there's ways, and if you got enough increased growth, you could do it. So most of the time, in his case, selection was the way to go. But I know there would be times in which changing the management would probably pay.
Is that the general view of everyone here? You would typically amend your selection program to fit your environment.
We did the opposite. We grazed the Suffolk ewes on irrigated pastures that were not hayed. They were just wild grasses. And we figured out that we had a worm problem really quickly. And I didn't want to deal with it anymore. So instead of selling all the sheep, we just changed around and we grazed them on dryland pastures, which we were lucky enough to have that. I know not everybody has that, but that's what we did. We didn't put them on the irrigated pasture anymore.
So in relation to that, I guess it defines for me whether you say environment includes management or you split out the M. Because nutrition, supplemented nutrition, or opportunities, as you say, to have alternatives within a given area, could be considered management, not environment. So if we're thinking topography, elevation, temperature as environment, feeding strategies given economics as management, now you got a three-way interaction, which is really fun.
But the way I think of it, this is important. Climate is, except perhaps the change that Dr. Brito raised, is a fixture of our location. We're not going to often shift our flock somewhere else. So we have to live with that. But we can manage around it if we have resources to do so. And that's a choice. Because the reason I'm raising this is because if this is an effort that we go through to develop strategies to allow you to pinpoint breeding values that may fit an environment, it's a whole change in how we go about doing a genetic evaluation. So if the result is changing management, given our climate, to make our animals fit, that's a very different approach than us saying, we're going to do all this cool stuff, which we would love to do, on creating reaction norms that helps you pinpoint that if I have this sheep in this environment, I expect it to do X. If it's in a different environment, I expect it to do Y. And so the issue is, are we creating a solution to a problem that you don't care about? Because that doesn't make sense. Is that clear, my question?
It does, and I'm not sure if we did the right thing, I'll be right honest with you. But we just couldn't, you couldn't take those kind of losses, I felt.
I guess you can just see here, for example, so what you want to do is you want the phenotypic value to be higher. So you want to increase performance. And then you have here, let's say the environmental value, and environment is everything that is non-genetics. So let's say that here is the lower or the amount of rain. So here is low rain, on the other side is high rain. So either you can, if your farm is located in a low rain area, what you want to do is, you will probably want to raise blue animals. If you're in a, let's say in a high rain, you may pick the blue animals. That's one strategy. The other strategy is, if you are here on the low rain, but you say, I like the orange animals, then you can select the orange animals to also increase their phenotype. So you can breed orange animals for also increasing their performance in that specific environment. So those are basically two options that we have.
I think another way to think about this, I mean, you're all seed stock producers. Think about your clientele. Maybe changing the environment to you makes more sense than selecting for different genotypes for your environment, but think about the diversity of environments that your clientele are purchasing rams. And maybe you can, identify rams that work better for them. If you're raising Suffolks in Michigan and trying to pair them to a ranged flock in California, maybe that makes sense. So there's different ways to think of it, but remember, we're all supposedly at the top of the pyramid making the genetic change and developing the tools for the rest of the industry.
We talk about selection and breeding programs. One of my thoughts is sometimes we have to let Darwin enter into effect. If you go back this morning to Ron's comments and the results of the breeder survey and the amount of interaction that different breed groups provided to their flocks. Our breed, we raised Rambouillet, had the lowest amount of interaction of the three breed groups. And maybe it's because we let Darwin take effect and we don't have inherent softness that Ron says Suffolks, and I agree, because we used to raise Suffolks, that Suffolks have. We create demand for human interaction by giving human interaction, and sometimes we have to bite the bullet. And sometimes it costs money to do that in the short run.